Okay, good morning, everybody. Why don't we get started? So how'd you like the first real nitty-gritty lab? Brains. What'd you think of those brains? Gross? Yeah? Was that a good thing? Yeah, okay, good. Did it help you to actually see it and manipulate it and tear the cortex? You thought that was helpful? Yeah. Okay, so that's what we're trying to do. And we're not going to go over memory for a number of different weeks. But, you know, we are two lectures away from your first exam. And I thought it would be useful to talk just a second about what helps your memory for material, like the stuff that we're going over in class. So it's a fair amount of material we've gone over. Lots of facts and names to remember for anatomy, lots of concepts, but also names for the electrical and chemical synaptic transmission that we've been going over. And I'll tell you the next two lectures is quite filled with different terminology to learn as well. So what's helpful for memory? What do you think helps your memory, just from a student point of view? What's going to help you? Yes? Repetition, perfect. We know that from both kind of psychological studies, but also repetition in our cellular models of learning and memory. You repeat something, you are going to remember it more. What else? Yes? What? Sleep. Very good. We're going to cover that in another lecture. Sleep and getting good sleep before the exam is critical. Also, if you got terrible sleep last night, you're not going to be able to really pay attention in class. That brings up another one, attention. If you want to try and gather the information that I'm talking about or any of your other professors are talking about, you must pay attention. No looking at the internet, even though I can't see what you're looking at on your computers. No focusing on other things. I'm not saying it's okay, it's not okay just to kind of take a little mental break. But you really, to pull the things that I'm telling you into long-term memory, you must maintain a good level of attention. Okay, so we have repetition, we have attention, we have sleep. Anything else? Any other ideas? Yes? Organization of information, perfect. That's another way to say associations. So associations with the things that you are learning now, with other things that you already know, are going to help strengthen those memories. And that's why I try and go back to different themes, and I try and kind of pick up on the things that we've talked about before to pull them into how they're relevant to what we're studying now. Because the more associations and links you have with something that you learn and with something that you've already learned, the stronger that memory is going to be. Did you have a new one? Mnemonics or acronyms, very good. So that's another form of repetition, but repetition in another way. The other last one that I'll bring up is emotional relevance. So we all have the phenomenon of, you know, traumatic event. You remember it really, really well versus, you know, 4 days ago, what did you have for breakfast? Who knows? Okay. Not that emotionally relevant, what you had for breakfast 4 days ago. But everybody can come up with one of those kind of lifelong haunting memories that's usually associated with something bad. It doesn't always have to be something bad. Something great you can remember as well. So, you know, if I got up and I slapped one of the students in the face, would that be memorable? Would you remember this lecture on drugs in the brain if I came up to you and slapped you right in the face? Would that be memorable? Yes. Okay. So I'm not going to do that, so don't worry. I'm not going to do that. But what you can do is to try and make some of these facts, perhaps the facts that are harder for you to remember, a little more emotionally relevant. I read this great book called Moonwalking with Einstein. Anybody hear of that book? It's about a journalist who decided to train for the Memory Olympics. And, you know, it's a bunch of really nerdy people that try and remember all, you know, that develop these techniques to remember. But it's actually amazing. They remember thousands of words or thousands of numbers, all in a particular order. And what they do is they use humor a lot. And they try and remember it by creating a story with these different sequences of numbers that are very humorous to you. That's the same idea of using emotional relevance. If it's funny, you remember it, okay? So just a little tip as we move into our preparation for exam 1, that will be Wednesday, October 5th. Oh, and let me just, one more reminder before we start the lecture is that you'll notice on the syllabus for October 3rd, it says evolution and use of animal models and in-class exam review, okay? So I recommend that you prepare for that by studying and being prepared for going over a review. It's going to be a little bit of a surprise. It's going to be a little bit of class participation, and it'll be fun. So come with the intention to kind of test out your knowledge on October 3rd in preparations for October 5th's first exam 1, midterm 1. Okay, so why don't we get started? So today's lecture is about really continuing our conversation about neural communication, neurotransmitters, drugs, and the brain. And we spent so much time over the last 2 lectures going over electrical and chemical transmission. So now you know this language of action potentials and how it turns from an electrical kind of communication to a chemical communication, but allows this basic kind of communication to happen in the brain. The majority of the communication that's happening in the brain happens using the currency of action potentials. But that's really kind of the black and white, the mugshot, as Chuck Close called it. Just the basics, you know. What we want to do today and over the next few weeks is turn this kind of basic representation of what's happening and give it a little bit more color, okay? And what we're going to do to give it a little bit more color is show you how different neurotransmitters and different receptors can play on this basic communication and create the kind of communication that is underlying not just, you know, a simple motor reflex response, but underlying our creative minds, our most creative selves. It can't happen with just simple communication like a reflex arc. We need a little bit more color. We need a little bit more to work with. And what we're going to talk about today are those specific kinds of neurotransmitters and receptors that add color to our basic action potentials. Okay, so neurotransmitters, you know what neurotransmitters are. Those are those chemicals in those little vesicle packets in the presynaptic terminal. When action potentials come down, calcium gets released, calcium inflows into the presynaptic terminal, and you get release of a neurotransmitter. But here I want to delve a little bit deeper and ask, what exactly defines a neurotransmitter? Well, there's specific definitions of what we categorize as a neurotransmitter, a substance that we characterize as a neurotransmitter in the brain. First, it has to exist in presynaptic action axon terminals. And typically what we do is we'll find a way to stain chemically for this particular neurotransmitter, maybe find antibodies to this neurotransmitter, and be able to look anatomically and localize it to the presynaptic terminal. Presynaptic cells, another defining characteristic of neurotransmitters, is presynaptic cells not only contain the neurotransmitter, but contain enzymes that synthesize that substance. So you have to both have the substance and have the machinery and the enzymes and the proteins that help build or make that substance within the presynaptic axon terminal. The substance must be released when an action potential reaches the terminal through that fusing of the synaptic vesicles with the presynaptic membrane. You have to have on the postsynaptic membrane specific receptors that recognize that substance that you think might be a neurotransmitter. So you have to have it there. You have to have something that makes it. You have to have receptors that recognize it. you have to show that application of this substance that you think might be a neurotransmitter produces changes in the postsynaptic potential. That is, binding of that substance on the receptor is actually doing something electrically at the level of the postsynaptic membrane. And finally, you have to show that blocking release of the substance prevents nerve impulses, prevents the propagation of action potentials, or EPSPs or IPSPs, to the postsynaptic potential. So all these different things have to be true for a substance to be called a neurotransmitter. And using these definitions, we've come up with a number of different categories of major neurotransmitters in the brain. I'm going to focus on two major categories, which means you need to know about two major categories. The first major category is actually number 2 on this list, amino acids family of synaptic transmitters. These are the biggies, the major excitatory and inhibitory neurotransmitters that we'll go over in a second. The second major category is number 1 on this list, the amines. This includes acetylcholine. We talked about this with Lowy and his experiment. Norepinephrine, dopamine, serotonin, and those are the main ones we'll go over. So amines, one subcategory, and we're going to start with amino acid neurotransmitters. Okay, why do we start there? Because those are our kind of bread and butter neurotransmitters. The major excitatory neurotransmitter in the brain is glutamate. It is one of the subcategory of amino acid neurotransmitters. Systems that use glutamate as its neurotransmitter are called glutaminergic. So glutaminergic transmission uses 3 major kinds of receptors. And I want you to know about these and know at least the fact that they are ionotropic. Why? Because they're going to come up again when we go over learning and memory. So the 3 major glutaminergic receptors are AMPA, kainate, and NMDA. These all refer to different drugs that activate these different specific kinds of receptors, and all of those receptors are ionotropic. I'm going to remind you what that means. There are also metabotropic glutamate receptors, just so you don't think it's too simple, but I will focus and I will ask you to remember and learn about the ionotropic neurotransmitters. I'll remind you that there's 2 major types of receptors. One are what's called ligand-gated receptors. They are ionotropic. Remember, a ligand, which is basically a key that fits a keyhole, comes in and it opens a channel that is then permeable to a particular ion, ionotropic. These are fast. We've talked about them before. And the second major kind of receptor is metabotropic receptors. They are indirect receptors, and they use a second messenger system. So this first receptor that receives the ligand or the neurotransmitter, another word for ligand, does not let any ions through. What it does is this receptor activates a second messenger, usually what's called a G protein, that then goes and activates an ion channel. So you can imagine this is a slower kind of neurotransmission, but there are multiple different steps, and there are multiple different steps that you can go in and intervene in this kind of metabotropic receptors. That is, drugs or other inventions can tweak this process. Okay, so while glutamate is the major excitatory neurotransmitter, the major inhibitory neurotransmitter in the brain that I'm sure that you've all heard about is called gamma aminobutyric acid, or GABA for short. Okay? There's another one that I'll also ask you to remember. There are 2 inhibitory neurotransmitters, GABA and glycine. They're both inhibitory and both amino acid transmitters, that first category on that list of neurotransmitter families. Now, we're going to focus on GABA because it is most common. This is an inhibitory neurotransmitter, and there are actually 3 different classes of GABA receptors in the brain. The first is called the GABA A receptor. We're going to come back to this at the end of the lecture. GABA A receptors are ionotropic, producing fast inhibitory effects via chloride channels. So is chloride high or low outside of the cell? High concentration or low concentration of chloride? High, thank you. Perfect. So chloride channels open. Chloride, which is negative, goes into the cell, and what does that do to the cell? Hyperpolarizes it. Perfect. So that's what the GABA A receptor. When GABA A is activated, you get a hyperpolarization of the postsynaptic membrane. Okay. What about the other 2 types of GABA receptors? GABA B receptors is metabotropic. It's slower, and it has inhibitory effects through neurogliaform interneurons. You don't have to know that, but just know that GABA B is metabotropic. And GABA C is another category of ionotropic channel, also associated with a chloride channel, but has different properties. Okay? So what are kind of drugs that can play with these GABA receptors? And it won't be so surprising to you that a major drug that plays with the GABA receptors that are, again, controlling inhibition in the brain is Valium. So Valium are potent tranquilizers, and they're also agonists of GABA. What does an agonist of GABA mean? What is it doing? Is it like GABA, or does it block GABA? It's like GABA. So you put a lot of Valium in your system, it's like activating all your GABA receptors. You are shutting things down. You are getting calm. That's exactly what happens when you take Valium. Alcohol, we'll talk about this more at the end of the lecture, also works on the GABA A receptors, and we'll go over that a little bit more detail at the end of the lecture. Okay, so that is the major excitatory and inhibitory neurotransmitters throughout the brain in terms of numbers. There are millions, perhaps billions of glutamate and GABA receptors, amino acid receptors throughout the brain. They are widespread throughout the neocortex as well as subcortical structures. But now I want to go into some of the most important other neurotransmitters. These are the amines, okay? And let's start with the one that we've already talked about, acetylcholine, the first major neurotransmitter that was discovered. If you remember Lowy and his experiment with the heart. So we're going out into the peripheral nervous system where he stimulated the vagus nerve. He got the heart to slow down, and he slurped up all the heart juice, and he put it on another heart, and he got that heart to slow down without stimulating anything, showing that something was in that heart juice that was coming out, we now know from the synapse, and that substance that they later purified and identified was the neurotransmitter acetylcholine. Acetylcholine and the enzymes that produce acetylcholine, which will identify cells that are what's called cholinergic, was mapped by looking at antibodies for the enzymes involved in acetylcholine synthesis. So again, cells that make acetylcholine, that use acetylcholine, are called cholinergic. Cholinergic nerve cell bodies and projections contain acetylcholine. So that's just a definition of cholinergic. Cholinergic nerve cell bodies and projections contain acetylcholine. Okay, where are the main centers for the synthesis of acetylcholine in the brain? The main location of the synthesis are 3 major areas. The first is called the nucleus basalis. It's right here. This is, as you know, a mid-sagittal cut of the human brain. Lots of acetylcholine cells, cholinergic cells, rest in this nucleus. A second structure right here, very close to it, is called the medial septal nucleus. Lots of cholinergic cells here. And the third one is pedunculopontine nucleus here. Now you can say see that these two major nuclei, nucleus basalis and medial septal nucleus, these are the cholinergic cells that are projecting throughout the neocortex. Up here, they provide acetylcholine to the neocortex. We're going to talk about what exactly acetylcholine is doing. Whereas this pedunculopontine nucleus is providing acetylcholine to the subcortical and brainstem structures. Okay, so that's acetylcholine and where it's synthesized, but that's only one half of the equation. What about the receptors for acetylcholine? What are they like and where are they? They're certainly located in these projection zones of the acetylcholine cells. There's two major types of acetylcholine receptors. You need to know about both of them. The first is called a nicotinic receptor. Most of the nicotinic receptors are ionotropic, and they are excitatory. Now, here you'll start to see the complexity. Most of them are. Some of the cells that are activated by actually a substance called nicotine, that's how they were identified, could be inhibitory. So there's always a little bit of wiggle room here, and it just points out the complexity of these systems. So what are some examples of parts of the brain that are using nicotinic acetylcholine receptors? And the example that you're most familiar with are muscles. All muscles in the brain that are being stimulated by the motor axons and the motor axon terminals, they use nicotinic acetylcholine receptors. And so including your heart, all of your muscles of your body, and paralysis can be induced with antagonists of nicotinic acetylcholine receptors, such as curare, which will completely paralyze an animal, just not be able to move at all, because they're blocking the nicotinic acetylcholine receptors in your periphery. The second major subtype of acetylcholine receptors is called muscarinic. These are metabotropic receptors, and again, can be excitatory or inhibitory. Okay? So you have nicotinic in the muscles, muscarinic can be excitatory or inhibitory. You also can have nicotinic acetylcholine receptors, the ionotropic ones, in the cortex as well. Okay. So, oops, it went the wrong way here. Okay, what are the functions of acetylcholine in the central and the peripheral nervous system? We talked about one already. The first is acetylcholine is associated with motor functions. You antagonize acetylcholine in the periphery, and you lose all ability for voluntary movement. It goes to your heart, and your heart stops beating. Okay? But it's also involved in the central nervous system in learning and memory. In fact, cholinergic neurons are highly, highly susceptible to death in Alzheimer's disease. In Alzheimer's disease, a lot of the, and sometimes all of the cholinergic neurons in the nucleus basalis and medial septal nucleus in particular, the ones that are going through the neocortex, they die. And there has been ideas that, or hypotheses that acetylcholine is critical for learning and memory function, and also very susceptible to Alzheimer's disease. So it's been implicated in learning memory as well as motor functions. Okay, second major neurotransmitter is another one that has two interesting complementary roles. Here, acetylcholine, motor function, learning memory. Dopamine, again, is another form of motor function, but also reward. Okay, so dopamine, abbreviated DA, is found in neurons in what's called the mesostriatal pathway. These originate in the midbrain, specifically in a structure called substantia nigra. Substantia nigra means black stuff, okay? And it's actually, there's a coloration there. So if you look, you can probably see it, not this week, but next week when you do the Make Your Own Brain lab. There'll still be some sheep brains there. And if you cut to the right part of the midbrain, you'll see a nucleus with black colored cells. You can see this in the sheep brain. With that, you will identify the substantia nigra, where all of these dopamine-containing cells live. This pathway, the mesostriatal pathway starting in the substantia nigra, is important in motor control. And loss of the dopaminergic neurons in the substantia nigra causes Parkinson's disease. So once you start losing these cells, you don't lose your ability to completely move, but you lose your ability to control fine motor movements. Well, so what's the most common characteristic of Parkinson's disease? How can you tell somebody has Parkinson's disease? Yes, shaky. So they can still move, but they have a shake. The other thing Parkinson's disease patients have is an inability to initiate movement. Once they get going, they're fine. They have a hard time starting the initiation of movement, but once they get going, it's fine. It's interesting. Mark Morris and his dance troupe he has a dance studio in Brooklyn, for many years has been working with Parkinson's patients doing dance therapy. So once you get them moving and once you get them dancing around, they're fine. They move great. And the music and the, just the practice of getting moving and staying in a flow helps them because they are losing their ability to initiate movement from the lack of dopamine in their substantia nigra neurons, or I should say the death of the neurons, the dopaminergic neurons in the substantia nigra. Okay, what about the second major pathway for dopamine? That is called the mesolimbocortical dopamine pathway, and this also originates in the midbrain in an area called the ventral tegmental area, VTA. These neurons in the VTA project to what's called the limbic system, important for learning, memory, and emotion. Have you all heard that term before, limbic system? Yes? Yes, okay, good. So limbic system, important for learning, memory, emotion, as well as the cortex. So dopamine in this pathway, originating in the ventral tegmental area, is involved in reward, reinforcement, and learning. And abnormalities in this pathway are associated with both schizophrenia and addiction. You can imagine that a pathway important for processing information about reward could be affected in addiction, where people get obsessed with any kind of reward, with a drug reward. And we'll talk about how some of these drugs are working directly on this dopamine system, or a monetary reward in gambling addiction, for example. Well, it's the same neurotransmitter that's so important for motor control in Parkinson's disease that's also critical for reward and reinforcement processing. Okay, where are these pathways? Here are the midbrain pathways, midbrain nuclei right here. Here's the VTA, more on the ventral surface of the brain, and right next door is the substantia nigra. And substantia nigra is going to the caudate and putamen, some of these subcortical structures that you'll look at a little more closely in 2 weeks in the lab. I know some of you identified the caudate nucleus already in the lab sections. And then here's the VTA going to the limbic system. Here's the hippocampus, important for memory. The amygdala is right in front of it, as well as the rest of the cortex. Okay, that's dopamine. Next, norepinephrine, abbreviated NE. This is released in 3 different brainstem regions. First and foremost, this is the most important one, is called the locus coeruleus. It's at the level of the pons. Okay, everybody identified the pons in the sheep brain dissections. The second location of norepinephrine-containing neurons is the lateral tegmental system in the midbrain. And finally, the dorsal medullary group. All 3 of these contain norepinephrine, but the largest location is in the locus coeruleus. Okay. These fibers, and these are called noradrenergic fibers, that is the axons coming from cells that synthesize noradrenaline. And these fibers from the locus coeruleus really project super broadly in the brain, all over the neocortex, as well as subcortically. The central nervous system has 4 subtypes of norepinephrine, noradrenergic receptors. All of them are metabotropic. Okay? So imagine the difference between the glutamate system and the noradrenergic system. The majority of all the glutamate receptors are ionotropic. They are fast. Once you get a signal, it is being conveyed in a very, very kind of strong and direct way. Noradrenaline is doing something a little bit different. It's a little bit more laid back. It's a slower process. All four subtypes of these receptors are metabotropic. No way for this information to get across as fast. And it makes sense because norepinephrine is what's called a modulatory system, whereas glutamate is helping convey information like vision to your brain. You want that to happen fast. Norepinephrine is modulating processes, including mood, arousal, and sexual behavior. So those things are happening at a more slower pace, as illustrated by the metabotropic receptors. Okay, and so here is the location. The major location that we're showing here is the locus coeruleus. The amazing thing about this system is that it's a teeny, teeny, tiny little nucleus right in the midbrain. This is, as you know, a mid-sagittal section. Here is a big lump here and a little lump here. What are these two lumps? What is the big lump? Located up or superior. Sheep brain had the same lump, kind of right behind the corpus callosum. Any guesses in the midbrain? Superior colliculus, thank you, perfect. And the little one? Inferior colliculus. These are the midbrain structures. And we go down here, of course, this is what? Cerebellum, and then right across from the cerebellum is the pons. Okay, so our locus coeruleus here is right kind of in the heart of the midbrain at the level of the, more like the inferior colliculus. Again, it's a very small nucleus, but all these critical cells are going out and innervating widespread brain areas. And attention and arousal is critical. You would fall asleep in all your classes if your noradrenergic system wasn't activated, at least a little bit. Okay, last neurotransmitter system before we start talking about how to tweak neurotransmitter systems. The last neurotransmitter system, you probably, even though you might not realize it, have heard a lot about. This last neurotransmitter is called serotonin. Its abbreviation is 5-HT. Cell bodies are mainly found in what's called the RAF-A nuclei. And again, its serotonergic fibers project widely. Serotonin is implicated in sleep, mood, sexual behavior, and anxiety. And for this discussion, we'll focus on mood. Who has heard of an SSRI? Raise your hand. What does SSRI stand for? Who? Yes. Yes, thank you. Is that what you're going to say? Yes, exactly. Perfect. Serotonin, SSRI, serotonin reuptake inhibitor. So what does that mean? The reuptake of serotonin is being inhibited by the SSRIs, okay? What do you think that would do? What do you think that refers to? Yes?
It blocks the serotonin from being uptaken, so later in your brain.
Say that one more time, the last part.
It's floating around in the synapses, so you have like an ecstasy type effect, right?
You have, so yes, you're blocking the reuptake. So tell me again, she just said you're blocking the reuptake. And what's happening in that synaptic cleft because you're blocking the reuptake? It's there. There's more there, right? Okay, perfect. Did you have something?
Say that it would keep sending the signal, so like, if it's taken back, if it's absorbed back, then
Right.
the signals will stop.
Exactly, exactly. Perfect. So she said that the signals, there's more signal going there because there's more serotonin in the synapse. And if that reuptake is working, that signal goes away because they suck up, they vacuum up that excess neurotransmitter. Remember we talked about house cleaning after an action potential comes in. You have all this neurotransmitter that was just released. If the next action potential is going to have the same effect, you need to vacuum that back up and take it away. And there were two ways that we talked about that could happen, the cleaning up. One are enzymes that break down the neurotransmitters. So they're there, but they become inactive in the synaptic cleft. And the second is there are specific reuptake mechanisms so that basically the active transmitter can get sucked back, vacuumed back up into the presynaptic terminal, and then repackaged and used again whenever the next action potential comes along. So let's go back to SSRIs. They are blocking reuptake of serotonin. Therefore, there's more serotonin hanging around. And what is the effect of SSRIs? Antidepressants. So you are improving mood right here. So more serotonin, more activity of serotonin. Not for everybody, but for a large population of people will increase your mood. Okay, so here are the complex set of RAF-A nuclei that go from the midbrain here to the level of cerebellum in the pons to the medulla down here. And again, these multiple different nuclei throughout the brainstem are going all over the brain to activate the serotonin receptors. And less activation in these can lead to depression. Perhaps it's the cells that don't produce enough serotonin. Perhaps it's a problem with the transmission of the serotonin. But the bottom line is using SSRIs that can pump up serotonin in your synapses can improve depressive symptoms. Okay, so we started with, this is a good lead-in to the next question. We were talking about a drug, a very, very commonly used drug that's tweaking our neurotransmitter systems. And so let's just be a little bit more specific here. What is the definition of a drug? So a drug is an exogenous substance, a substance that isn't normally found in the brain, that has an effect on behavior, mood, emotion, activity, or health. Okay, very broad definition. Second, some are natural substances and some are man-made, these drugs. So many of the drugs have been derived from plants that we've known about for ages and ages because our early ancestors used these medicinally, chewed on them to get the active substances out, smoked them to get the active substances out, and we just are a little bit more sophisticated so we can extract them with complex chemical reactions. And as you know, drugs can be both beneficial as well as harmful. So drugs used in the context of medicine can be fantastic and improve so many different medical conditions, but they can also be abused, which can be very, very harmful. Okay, and many of the drug effects act via the neurotransmitter systems that we've just been going over, and specifically the neurotransmitter systems at the level of the synapse. So we talked about one way in which this drug, SSRIs, how these SSRI drugs are targeting one aspect of normal synaptic function, and they're inhibiting reuptake. Okay, so imagine, try and come up with a drug, and try and come up with your favorite drug that affects one form of one aspect of synaptic transmission, either presynaptically or postsynaptically. Let's start presynaptically. So somebody give me a kind of made-up drug that's going to affect some kind of process in the presynaptic terminal, and tell me what it's going to do. Is it going to enhance that transmission, or is it going to block that transmission? So what are some of the processes that we talked about in the presynaptic terminal? Yes? Did you have a? No. So what happens when an action potential comes down the axon? And what happens at the postsynaptic terminal? What gets activated? Vesicles. Okay, how do vesicles get activated? Calcium, great. So what would happen if you blocked those voltage-gated calcium channels? Vesicles wouldn't fuse. You would actually block all action potentials if you blocked the voltage-gated calcium channels. Some classes of drugs can target those voltage-gated calcium channels. That would be devastating. You don't have voltage-gated calcium channels. You don't have any action potentials coming down. Okay, so great, you got us started. We have calcium, we have action potentials coming down. It is depolarizing the voltage-gated calcium channels, and calcium comes in, and we get fusion of those vesicles at the presynaptic terminal. What if you had a drug that blocked fusion of the synaptic vesicles? You can get calcium flowing in just fine, but vesicles can't fuse, okay? What would happen then? Yes? Exactly, same effect. You would block all neurotransmitters. So what if I now had a drug that enhanced, that just released all neurotransmitter? You're all synapses. I'm just going to put a drug on you that releases all neurotransmitters. What would that do? Yes? Right, it would multiply the effect, but in a really bad way, right? So we have, in normal brains, you have specific reasons why you're getting the fusion of the vesicles with the presynaptic membrane. But if you have some drug that gets put on and then suddenly all of the vesicles are fused, you get just chaos. Okay, so let's look at this in a systematic way. The point was that we've gone over all of these steps of how neurotransmitters are released. You have action potentials coming down the axon. Here is the voltage-gated voltage-gated calcium channel. Calcium comes in. You have synaptic vesicles that are fused. And the point is that all of these different steps can be targeted by different drugs. So you could now imagine yourself as the head of a pharmaceutical company, and you have a particular problem that you're trying to address, not in all synapses, but let's say in cholinergic synapses. Let's say in cholinergic synapses in the frontal lobe. Can you design a drug that will go in and either enhance synaptic vesicle release in those particular subcategories of cells, or can you go in, let's say they're more overactive, can you go in and just moderate that activity in a specific way? That is part of the strategy that drug companies use. They need to know these mechanisms like the back of their hand and try and go in a clever way and manipulate it such that the patient is helped. So you can see all these different steps. Synthesis of neurotransmitters can be affected. Conduction of the action potentials can be affected by drugs. Axonal transport of the synthesized neurotransmitters that can be synthesized in the cell body and then transported down to the axon terminal. Release of synaptic transmitters, storage of the transmitters can be affected, modulation of transmitter release by presynaptic receptors, and inactivation, the SSRI kind of strategy, inactivation of transmitter reuptake. And that's only on the presynaptic side. Drugs can also modulate different mechanisms happening on the postsynaptic side as well, and that is where we're seeing most of the receptors, the transmitter receptors. You can block the receptors, you can stimulate the receptors with receptor agonists. You can inactivate the transmitter, so here is a kind of mechanism in the synaptic cleft. You can alter the number of postsynaptic receptors by stimulating receptor kind of synthesis on the postsynaptic side. Lots of different ways that drugs get in there. Yes?
How do doctors know what medication to prescribe without knowing what exactly needs to get, like which part needs to get modulated?
Yeah, that's a really good question. And sometimes the drug effects are by chance. So doctors are treating certain sets of symptoms, and they find actually this drug is also helping something else. I didn't even know that. And then drug companies can go back and say, well, what is it doing in this system that we know a little bit about, and can we see what effect that is? That's part of the way that we go in. The other part is through basic research, knowing that, let's say you have a learning and memory problem, and let's say we want to try and develop a drug to enhance your learning and memory. This is a very, very hot topic. There are so many startup companies trying to come up with the memory-enhancing drugs. What you do is you go back to the basic literature and you learn all you can about the systems and the transmitters and the receptors that we know are involved, and then try and go design your designer drug that will enhance your memory without making you, super memory person or having horrible side effects that you can't even function. You can't take advantage of that drug, so or of that effect. So it's this interesting combination of knowing the basic research that you study in slices of mammalian, just rat brain, and going and talking and interacting with the patients to see what is this drug doing. Yes, question? Would side effects just be learned through trial and error? Yes, and there is a very, very strict system in process to make sure that you can't just test any old drug. Let's just try this, I don't know if you saw Rise of the Planet of the Apes, but scientists do not go home and try the drug on their father, okay? They just don't do that. You're not allowed to do that. And so what happens is there's a series of steps you have to go through. You try it on animal trials and on animals to make sure that it's fine. If it passes a lot of stringent tests, it gets to first-order tests where just a few people can be tried just to make sure there's no long-term or short-term effects. But it goes through a process like that. Sometimes later on, you didn't even realize there was an effect, but it could be a long-lasting effect. People didn't realize it, and that happens as well. But there is a systematic way to test that side effects are not devastating. Any other questions about drugs? Drug testing or drug development? It's actually a fascinating topic, and some of the brightest people are there trying to tweak this amazingly complex system to improve cancer treatment or improve recovery from damage, and do that in a systematic way that doesn't have a lot of side effects. Okay, let's go on to drugs. Oh gosh, it's late already. So many drugs are ligands. Ligand is just a general word for that key, that thing that is activating a receptor. Any chemical substance, amino acid, that is activating a receptor in the brain is called a ligand, okay? Many drugs are ligands that act upon specific receptor molecules. And these drugs can be very specific, or in most cases, they're quite messy, and they act on a number of different receptors. And that's where you get side effects coming in that may be positive or negative. Now, because receptor subtypes have different localizations and functions, drug actions can have widely varying effects. So we talked about agonists and antagonists. Drugs can act as agonists, and agonists are defined as molecules that act like the transmitter at a receptor, like the endogenous transmitter at the receptor. And if you act like the transmitter and activate what you're supposed to activate, what's normally activated, you're called an agonist. But if that same molecule fits into the receptor. It's still a ligand, fits into the receptor, but it interferes or prevents the action of the normal transmitter at the receptor. They are called antagonists. Okay, so very important concept. We talked about this before. Again, ligand is the general term. Anything that fits into the receptor is a ligand. A drug is a ligand. A neurotransmitter is a ligand. So both a neurotransmitter and a drug can be either an agonist or an antagonist. I should say the drug can be either an antagonist or an agonist. And the neurotransmitter is always normal. It works as normal, which is the agonist. So let's talk about the concept of competitive ligands and non-competitive ligands. Competitive ligands bind to the same receptor site as the endogenous neurotransmitter. Okay? So you just have to ask yourself, is this the regular neurotransmitter site or is it a modulatory site? If your ligand is activating a modulatory site, it's called a non-competitive ligand. And the modulatory site, when bound by compound, alters the receptor's response. Okay? So here is the neurotransmitter on its typical receptor. This is the red little tooth-like looking thing. And when a drug comes and activates that site, this could be an agonist, binds to the receptor site and activates them. Drugs that are agonists mimic the endogenous ligand. So let's say this is the agonist, and we know it's an agonist because it's doing the same thing. It's opening this channel. Here the channel is closed. When the neurotransmitter is on, the channel is open. And when the drug agonist is on, the channel is open. Okay. Now let's talk about the antagonist blocking the drug. A competitive antagonist will vie for the same exact receptor slot as the neurotransmitter. So here's the red thing being popped off, and this competitive antagonist is occupying this site, but it does not allow this channel to open. That is this antagonistic function. But this is the competitive antagonist. The non-competitive antagonist is binding not to the receptor site, but to a modulatory site. So here, the normal neurotransmitter, the red tooth, is binding right here, but this non-competitive antagonist is binding to the modulatory site. But guess what? That is still causing the closure, keeping this receptor or this channel closed. That is two different ways that antagonists can work, through a competitive mechanism or a non-competitive mechanism. Okay, so let's talk a little bit and add yet another level of complexity here. So we started with just simple action potentials. Then we added on different flavors of neurotransmitters. And then we added on different kinds of receptors for the different kinds of neurotransmitters associated with different kinds of functions. And now we're going to talk about the specific efficacy of those individual kinds of neurotransmitters or drugs. And for that, we focus on the topic of binding affinity or affinity. We're talking about binding to the receptor. And that is the degree of chemical attraction between a ligand, that key, and a receptor. The efficacy, or it's also called intrinsic activity, is the ability of a bound ligand to activate its receptor. Okay? And so let's look at just very nice visual illustrations of drugs that either have a high binding affinity or a low binding affinity. Here are these big square things are receptors, and only on this right-hand side here, that is the binding site. Okay, so this is not bound to a receptor, but here, with this much drug in the substrate, you get 2 of these 4 receptors being bound by drugs. Okay? Now let's compare that to this other drug. There's much less of this drug in the substrate, but with much less drug, you still get 2 out of the 4 receptors that get bound with the drug. That tells me that this yellow drug has a higher binding affinity than the blue drug. Does everybody get that? Pretty simple. Okay, good. Higher affinity, lower affinity. Drugs can have high affinity or low affinity. The other important concept in this kind of pharmacological aspect of drugs in the brain is a dose-response curve. And that is a graph of the relationship between drug doses and their effects. The drug dose-response curve is a very, very common tool to understand pharmacodynamics and the relationship between drugs and their targets and drugs and their effects. Okay, the other terminology that I need to tell you about before we get to the next graph is called the effective dose, the ED. And the ED50 is the effective dose where 50% of its effect can be seen. Let's show this, let's look at this in more detail in a typical what's called dose-response curve, DRC. A dose-response curve plots the response of the drug, whether it's for how much you get a fever down, or how much you stop coughing, or how much you change some other behavioral variable. And the response of that drug, behaviorally, is measured as a function of the dose of that drug, a log of the dose. But this is basically dose and response, dose-response curve. The effective dose, and you plot, at this dose, I'm going to see what the response is. And you go higher and higher and higher, see what the response is. And here we see there's very little response down here. There's a steep curve, and then the response doesn't keep going up even with, very high doses up here. And the effective dose, as I said, is that dose level. How many milligrams per kilogram here that you give that gave what you showed was half of the maximal effective response, okay? A drug that has comparable effects at lower doses is more potent. You don't want to have to be taking a whole gallon full of drug to get the response. And we compare drug efficacies by evaluating their maximal responses. So here are two nice examples. These are both dose-response curves. Here's the dose, here's the response, here's the dose and response. And here we're showing two different types of responses. This is the dose-response curve of two drugs that have the same kind of response. And you can see that drug A has the same 50% effective dose, the ED50, at a much lower dose than drug B. So which drug are we going to pick? Choose A. Okay, this is a different kind of relationship. Here is drug A again, but now we're testing it against a different drug B. They should have really labeled this a different name, but I got this from the book, and you can read about it all in the book. You can also evaluate not just the effective dose, but the maximal response effect. You want a drug that has the strongest possible effect. And here you see that drug A has a much higher, greater maximal effect than drug B, no matter how big the dose is. So obviously, drug A, again, wins, not only because of its lower dose, effective dose, but its higher maximal effect. Okay, and finally, the last concept before we go to the movie is the therapeutic index, which is the separation between the effective dose and a toxic one. So here is, so obviously toxicity is a critical issue in pharmacology. And what you're looking at is the doses where you get a good effect and the dose of a particular drug where you give so much you actually kill the animal. These can be, these are all tested in experimental animal situations. You want the difference in dose between the ED50, the effective dose 50, and the LD50, that's a bad one, the lethal dose, 50% lethal dose, to be as wide as possible. So here's drug A again, here's drug B. You can see that drug B has a very narrow therapeutic index. That is, you can, just with a little bit of a change in the dose, go easily from the effective dose to the lethal dose. That is dangerous. This is much safer. So these are some of the basic things you evaluate in drug efficacy. Now, in the next 15 minutes, I just want to turn to what I thought was a really good movie because it gives you a 3D kind of appreciation of drugs interacting with synapses. So we're going to talk about different classes of drugs from this movie. And then I'm going to tell you what you need to know from the movie. Okay, let's see. Great. Here we go.
Now that we know how neurons communicate with each other, we can examine how this communication is affected by various drugs. The brain is a highly interdependent organ. Small actions at specific places can have global effects, changing our feelings, moods, and perceptions. Scientists have taken advantage of this interdependence by developing drug therapies to treat brain disorders. Some people, however, take drugs even though they are not sick, simply to change the way they feel. Many drugs, both therapeutic and abused, are natural substances derived from plants. Let's start with the tobacco plant. The active compound in the tobacco plant is nicotine. Somehow, the nicotine in tobacco smoke affects the brain to change the way one feels. In fact, if nicotine is removed from tobacco, people won't smoke it. How does nicotine get from the tobacco into the brain? The real answer is through the bloodstream. When a cigarette is lit, the burning tobacco produces smoke, which is inhaled by the smoker. This smoke contains nicotine, which has become vaporized by the intense heat and released from the tobacco. With each drag of the cigarette, the nicotine travels along with the smoke into the lungs, where it easily diffuses into the blood vessels lining the lungs. These blood vessels then bring the nicotine to the heart. As the heart pumps, the nicotine is carried through the bloodstream directly to the brain. As they enter the brain, the blood vessels branch out into capillaries, carrying the nicotine within them. Normally, these capillaries carry nutrients to the neurons. Just as it moves from the lungs into the bloodstream, nicotine easily diffuses through the walls of the capillaries and travels to the spaces surrounding the neurons. As you will recall, many neurons contain the neurotransmitter acetylcholine, which is released from the nerve terminals into the synaptic space. Acetylcholine binds to receptors on the other side of this space to open ion channels that generate a current across the membrane of the receiving cell. Nicotine competes with acetylcholine to bind to the acetylcholine receptor. It wins, and then opens ion channels to let sodium ions into the cell, just like with acetylcholine. But there's a lot more nicotine around than acetylcholine, so a much bigger current is produced across the membrane. This bigger current causes increased electrical impulses to travel along certain neurons. With repeated smoking, the neurons adapt to this increased electrical activity, and the smoker becomes dependent upon the nicotine. This adaptation is illustrated in the caudate nucleus, an area of the brain that controls voluntary movement. Without the nicotine, impulses aren't maintained at the levels they were previously. As a result, some smokers experience hand tremors between cigarettes. These smokers' tremors may be hard to see because smokers hide them by smoking another cigarette. The tremors may be a sign of withdrawal, but they will go away when the smoker gives up smoking for good. The coca plant is another source of a mind-altering drug. The active ingredient found in this plant is cocaine. As with nicotine, cocaine acts on the brain by first getting into the bloodstream. When cocaine is snorted as a powder, it dissolves in the mucous lining of the nose and enters the bloodstream through the nasal blood vessels. With repeated snorting, the capillaries in the nose can become damaged.
Looks like Avatar, doesn't it?
Once in the blood, the cocaine travels to the heart where it's then pumped directly into the brain. If cocaine is smoked, it reaches the brain even faster. The faster the cocaine reaches the brain, the more likely it will be abused. Like nicotine, cocaine diffuses easily through the blood capillaries in the brain to reach the neurons. However, once in the brain, cocaine works very differently from nicotine. It acts on the terminals of dopamine neurons. Let's review what happens at these terminals. Dopamine is released from vesicles into the synaptic space. Once dopamine binds to its receptor, it activates the coworker G protein to generate cyclic AMP. Meanwhile, uptake pumps on the terminal membrane pump dopamine molecules back inside the terminal. This keeps the dopamine from accumulating in the synaptic space and prevents the dopamine receptors from being overactivated. Now, when cocaine is present, it too binds to a spot on the uptake pump, but in doing so, it somehow prevents the pump from working. The result? More dopamine in the synaptic space. With all the extra dopamine around, more dopamine receptors become activated. Eventually, more cyclic AMP is produced inside the postsynaptic cell, leading to larger membrane currents. With repeated use of cocaine, some neurons actually become more sensitive to its actions, resulting in potentially fatal effects. Cocaine is very effective at sensitizing neurons to electrical excitation, even after only one exposure. When neurons are sensitized by cocaine, a person can develop seizures similar to those suffered by epileptics. Even though cocaine seizures happen only rarely, they may be accompanied by an irregular heartbeat, which usually results in death. There is no way to predict if any one individual will suffer a cocaine seizure, whether it's their first try or after they've tried it several times. Other psychoactive agents are found in the marijuana or cannabis plant. The main active compound in marijuana is tetrahydrocannabinol, or THC, but there are actually several hundred active compounds. Marijuana is usually smoked so that the THC gets to the brain quickly. When the marijuana cigarette is lit, the burning cannabis releases THC into the smoke in the same way that burning tobacco releases nicotine. Inhaled with the smoke, THC diffuses into the blood vessels lining the lungs. With each beat of the heart, THC is pumped straight up into the brain, where it distributes throughout. Like nicotine and cocaine, THC diffuses quickly out of the capillaries into the spaces surrounding neurons. As the THC approaches neurons, it targets specific receptors located on the cell membrane. Scientists have recently discovered chemicals within the body that normally bind to these THC receptors. Like the dopamine receptor, the THC receptor also requires help from the G protein to perform its work. But in this case, when THC binds to its receptor, it signals the G protein to shut down the production of cyclic AMP. Without the proper amount of cyclic AMP, membrane currents cannot be generated. This disrupts normal brain function in those areas where the THC receptors are found. More than in any other brain structure, THC receptors are highly concentrated in the hippocampus. The hippocampus lies deep within the brain. There is one in each side of the brain, or hemisphere. The hippocampus is a structure important in learning and memory. Because of THC's ability to disrupt the function of the hippocampus, many people find that marijuana use causes the loss of short-term memory. With repeated use of marijuana, learning becomes disrupted as well. THC can also cause a change in perception of one's surroundings, similar to hallucinations produced by other drugs. Such changes in perception are produced by the actions of THC within the cerebral cortex. As we have begun to appreciate, the brain is an enormously sophisticated organ. The same features that give it extraordinary power and a...
Okay, so let me, in the spirit of repetition, helping you say things, we're going to remember things. We are just going to, I'm going to go over this again, but I just want to give notice to you that there are a list of things that you're going to have to remember from this movie. You're not going to have access to this movie. These were basic things. You should have be able to assimilate these, the effects of nicotine on the nicotinic receptors and side effects in terms of stability. Cocaine, very interesting. It is blocking reuptake. What other structure, what other drug that we talked about that blocks some sort of reuptake? SSRIs. Similar to cocaine, but this is working on the reward system. That means a little bit of cocaine is like getting massive amounts of reward at the same time. That's why it is so, so addictive. It's working on a specific system and increasing that. And then marijuana for memory. So we'll go over this again and see you on Wednesday.